Hello guys, and welcome back to Excel Academy. So we are doing biological membranes and going to continue with the active solid transport. So in the previous, we have talked about the different types of active transport, and of which we have also mentioned about the P type, F type, V type transports. Right now, today we are going to discuss about the important one. As I have mentioned before, also that ABC is really playing a very important role in the case of the MDR as well as in the case of the cancer, right? So what does MDR stand for? Any idea? MDR stands for, in the case of bacteria?
Multi-drug resistance.
Correct. It is the multi-drug resistance. Now, if you see nowadays, there is something called AMR project also going on. अगर कभी सुना हो ध्यान दिया हो in TV news or newspaper and all, they talk about the government funded AMR. AMR is your antimicrobial antimicrobial resistance. Okay, so इसका क्या problem आ रहा है? That nowadays with the changes or in changes in the lifestyle as well as increase in the pollutants, okay, and many other things, it all eventually resulting in making your bacteria so resistant towards everything. ठीक है, तो अभी हम जब मल्टी ड्रग रेसिस्टेंस बोलते हैं, तो इवन इफ आई गिव अ कॉम्बिनेशन ऑफ 5 to 6 ड्रग्स in order to kill the bacteria in bacterial infection, the bacteria are so resistant to these that they are able to survive as if there is nothing. Okay, so there are two ways by which a bacteria can overcome the action of the drugs. So एक mechanism होता है, so in both the cases it will be known as multi drug resistance. Okay, now, the first mechanism is, we'll come to all this thing eventually. I'm just giving you the gist of the story, like how the bacteria are achieving this multi-drug resistance. So, एक हो गया आपका ABC transporter. Okay, so as I mentioned before, so on the bacterial membrane surface is there. So these ABC transporters are there. So the bacteria understands that the foreign substance has come inside. Okay, let us say that it is a toxin. So when the bacteria understand that by mistake the toxin has come inside, they make it into a vesicle sometimes and they exocytosis. They do the exocytosis. Okay, if possible. If this is not possible, the other thing is using the MDR, or sorry, not the MDR, sorry. It is called the ABC receptors. That is your ATP binding cassette transporters. Obviously, there is a domain where ATP is going to bind. Correct? Now, in these ABC receptors, what they do is they are going to take this toxin and pump out. So, as I mentioned before also, the concentration of the drug is going to be high here, right? आपने बाहर से इसको apply किया है, so the concentration of the drug is going to be high here, and the concentration over here inside the cell it is going to be less. Correct? So the bacteria needs to pump from its inside to outside. So ये क्या जा रहा है? Against the concentration gradient, right? So obviously it is going to need the ATP. This is a basic mechanism behind ABC transporters, and this is the basic mechanism behind any active transporters. Correct? Are you able to follow?
Yes, ma'am.
Yeah. ये एक पहला मैकेनिज्म हो गया. Now, if I talk about second mechanism by which the bacteria is able to become resistant to the drugs is by cleaving the drugs. Okay. Now, how it is possible? So, अगर कोई भी drug है, okay, so that particular drug is going to have its maybe the organic or inorganic component, right? So inorganic and organic component will be there. So अब हम क्या कर सकते हैं? The bacteria will release certain enzymes which will break the drug molecule. ठीक है? एक बार जब वो break होके different components में divide हो जाएगा, so what will happen? It loses its mechanism, right? The mechanism of action. अभी वो किसी काम का नहीं रहा. So एक ये तरीका हो गया. ठीक है? So either they can release or synthesize enzymes which can detoxify the drugs, or you can have more expression of ABC transporters. Is this much clear to you, Shruti? The two mechanisms by which the bacteria are becoming multi-drug resistant in today's scenario. Yes. Yeah. Now there are few more reports. Okay. There are few more reports which talks about, suppose a bacteria is there, and what they do is they take this toxins. Okay. If suppose, see, कैसा है कि हर एक बैक्टीरिया के पास ये सारे मैकेनिज्म नहीं होंगे. So different bacteria will have different mechanisms. So what are the report says that if this drug comes inside of the cell, they will cover it with a vesicle and keep on storing in one area. ठीक है. इट जस्ट लाइक किसी एक घर में आप एक जगह पे जो चीज आप यूज नहीं करते आप उसको बस स्टोर करके साइड में रखते जाते हो सो दैट इट इज नॉट अफेक्टिंग द रेस्ट ऑफ द सेलुलर फंक्शन एंड दिस इज वन मैकेनिज्म बट इवेंचुअली व्हाट विल हैपन अगर ये कीप ऑन ऐड होता रहेगा देन द बैक्टीरिया माइट डाई बिकॉज ऑफ जब आप कोई गार्बेज ज्यादा दिन तक रखते हो व्हाट हैपेंस इट स्टार्ट्स डिग्रेडिंग एंड it is going to become toxic eventually. So that time the bacteria might die, but that particular generation is going to survive. So this is the third mechanism. Okay. Now let's talk about the ABC transporters now. Okay. So it is widely distributed in all the phyla. It consists of two transmembrane domains. We'll talk about this. And each transmembrane domain has got 6 membranes spanning alpha helices. So total अगर कोई आपसे पूछे like ABC transporter जो है, how many alpha helices are there? Then you should say that it is 12. Okay. Now it is very specific to their substrate and it possesses 2 nucleotide binding domains. Okay. So in the case of prokaryotes, ABC transporters they work as the importers and exporters. तो ऐसा नहीं कि हमेशा टॉक्सिन को ही बाहर फेंकने का काम है. There are many other things which is required by the bacteria, so even that can be taken in. Okay. For example, like ions, sugars, peptides, phospholipids, vitamins, polysaccharides. It is also involved in the RNA translation and DNA repair mechanism. Okay, so this is how the structure of ABC transporters look like. Can you see? So, how many are there? 12 transmembrane alpha helices are there. Okay, so this is one transmembrane domain, this is second transmembrane domain. And at the towards the C, you can see that the first nucleotide binding domain is there, the second nucleotide binding domain is there. So as far as possible, Shruti, try to remember the structure. Okay, जब आपको structure याद रहता है, then you can remember the mechanism also. Okay. Now let's talk about like how it is functioning. Now, what is the first step? So always remember, अभी इस case में क्या है that both the N terminal as well as your C terminal is towards the cytoplasm. Okay, एक ये चीज भी ध्यान रखें. Now, look at the mechanism here. So what is the first mechanism? Whatever the drug is there, it is present inside, correct? It goes and binds to your binding pocket of your ABC transporter. Now, just imagine this entire thing over here and this entire thing over here. Okay? Now, look at over here. So, अब आपके पास molecule है, this molecule goes and binds its binding pocket. Now, what is happening? This is going to get open only when the 2 molecules of ATP is coming and binding to the nucleotide binding domain. Now, what happens when it binds? Can you see? Both the domains are coming together. That results in the conformational change. So, what we have seen till now is that whenever ATP binds, there is always a conformational change. Okay? So, this conformational change results in the release of the molecule outside. Then, followed by ATP hydrolysis. So this ATP hydrolysis will bring your ABC back to its resting stage. Clear? Shruti, do you want me to repeat once again? Is the mechanism clear? This is
yes, yes.
compared to what we had discussed before, right? Yeah. No doubts here. Simple. Molecule आया bind हुआ binding pocket में, then ATP came, molecule got released because ATP binding resulted in the conformational change. Now, once the molecule was out, then ATP hydrolysis happened and the ABC transporter came back to its normal state. Okay. Now, this is the same thing. This is a different representation, but here we are showing the NBD, nucleotide binding domain. So you can see. First domain is there, second domain is there. Okay, this is a transmembrane domain 1 and 2, nucleotide binding domain 1 and 2. ठीक है, तो अभी हमने ये देखा ना, जिस bind होता है बाहर जाता है. ठीक है. Now, look at over here. So this is a coupling helices. इसका मतलब क्या? The connection between the transmembrane domain and the nucleotide binding domain. So this is that. Okay. Now what do you see inside? कहाँ पे आके आपका ATP bind हो रहा है? So we say that nucleotide binding domain, but ATP आके कहाँ bind हो रहा है? Okay. So this is the arrangement they have shown. So this is your ATP. This much I think I should take another color once again. Huh? Yeah. So this is your ATP. Okay. Now, these are the other motives and all which is helping in the proper binding of the ATP. Now, again, what you need to remember here is the signature motif LSGGQ. Again, these are the amino acids, correct? So, if you need to, I don't think this question is gonna ask, but if the question comes like how the ATP is interacting or resulting in the hydrolysis or opening of the ABC transporter, then you just have to understand that when ATP binds over there and makes the interactions with the signature motif, okay, then the TMD, okay, they come together. ATP bind हुआ अपना NBD में यहाँ पे what happens? There is a conformational change which makes the TMD to come together. तभी तो हमने इसको couple किया हुआ है, right? With the NBD. So when NBD में ATP bind होता है, there is a change in the NBD which brings out the change in the TMD. So eventually resulting in the opening of the ABC transporter. Okay. Now the example. So here we are going to talk about the MDR. So MDR1. So one example.
Ma'am, what was the switch histidine
there
in the picture before?
So switch histidine means it will make sure that the interaction is proper. See, this is what, na? Even if I'm not saying, you should understand that. ये क्या है आपके? What are these? Triple P.
The triphosphate of
Correct.
ATP.
And phosphates are negatively charged. Correct. Histidine is what you are positively charged.
Okay.
Amino acids. So they are making interactions
Yeah.
to ensure that the correct conformation ATP has bind to the NBD. Okay.
Okay, it was called switch, so I thought might be something.
So that is what और भी उसमें histidine होंगे. This NBD is what? It is protein only. So obviously there is going to be so many histidine. Just to specify one, they give the answer. They give the naming like this, like switch histidine and all. अभी क्या है? Like if you refer to what I do when I make the presentation and all, so I make sure that I collect all the information from different text references. Correct. So each text reference is going to have their own, what to say, the nomenclature and all. Little difference you will find. चलेगा? अभी clear है?
Yes.
Anything else? Do you just want to just go through one by yourself? अभी जो हमने discuss किया, just go through all these things. Now, A loop means when you say, so it makes sure that the adenine part of your ATP binds there properly. P loop, yeah, Walker A. So they are nothing but just making sure that the ATP is bound properly. Okay.
Okay, ma'am, clear.
Anything else?
No, ma'am.
Okay. Okay. Now we'll go with the example. Okay. Multi-drug resistance. So it's a kind of P protein, P glycoprotein. Now, when I say P glycoprotein, what is the meaning of it? P glycoprotein मतलब क्या होता है?
Phosphorylation related.
You tell me. Guess नहीं.
It involves phosphate.
Nah. What else? So P is not always phosphate related. It can be something else. Okay, so we are talking about ABC. ABC क्या करता है?
It transports.
It pumps out, right?
Yeah,
It comes out. So
plasma
that
membrane.
is, it's okay. I mean, it's not like anyone is gonna ask you कि P से pump ही क्यों है, P से faucet क्यों नहीं है, ऐसा कोई कुछ पूछेगा नहीं. Just that, the people, the scientists who were working and all, जिसको जो सही लगा, they give the nomenclature. Okay, so it is one of the drug transporters. Okay, so P is like for the pump glycoprotein because there are many glycoproteins. So they are found in the intestinal epithelium cells, capillary endothelial cells of the blood-brain barrier, hepatocytes, etc. The main function is associated with the secretion of steroid aldosterone and transport of phospholipids and cholesterol. Along with that, they transport drugs such as colchicine, vinblastine, and quinidine, etc. So, where have you heard about the colchicine and vinblastine? Yes, quinidine. What do you know about colchicine? What do you know about colchicine? ये बहुत बेसिक है, आपने प्रैक्टिकल अगर किया है तो आपको मालूम होगा. Yes.
1 second, ma'am. I can't remember right now. 1 second.
Anyone? I mean, Tanvi, Akya, I have you joined. Have you heard about colchicine? कहाँ पे सुना हुआ है?
Even I can't remember now. Like, I'm trying to remember, but I'm not
Okay,
getting it.
Tanvi, would you like to try?
Wait, was it gout?
Was it? क्या? I couldn't hear.
Gout?
Repeat.
Colchicine. Was it gout? For gout?
Gout. नहीं बाबा, बेसिक इन योर लैब. मैं तुम्हें डॉक्टर का नहीं बता रही. बेसिक इन योर लैब. लैब नहीं तो ये हमने 12th स्टैंडर्ड में तो डेफिनेटली पढ़ा है बायो में. ओके, सेल साइकिल हिंट. ठीक है, I guess you guys can give up now. I'll tell you the answer. Colchicine, vinblastine, all these are nothing but they are the compound which is actually protein. Okay, so not protein. So they are going to block your mitosis. जब हम लैब में कोई एक्सपेरिमेंट करते हैं एंड वी वांट टू स्टडी द डिफरेंट फेजेस ऑफ द माइटोसिस, ठीक है, प्रोफेज, एनाफेज, मेटाफेज, ये हम सब कैसे पढ़ पाएंगे? When you treat the cells with the colchicine and vinblastine, so what will happen? That is the time when you see that all these mitotic spindles जो हमारे रहते हैं, they get blocked because it prevents the microtubular proteins. Okay, it prevents the mitotic spindle formation. So what will happen? तब आपको मेटाफेज में ऐसे आपके क्रोमोसोम्स दिखते हैं in this stage. So what is happening? The depolymerization of mitotic spindle is prevented. तो जिसकी वजह से मेटाफेज में आपको ऐसे अलाइंड क्रोमोसोम्स दिखते हैं. नहीं किया लैब में कभी एक्सपेरिमेंट्स टेकिंग द अनियन पील? आई एम श्योर यू मस्ट हैव डन. ठीक है, याद नहीं होगा. एनीवेज, इट्स ओके. सो दैट इज योर colchicine. ठीक है. Now coming to the vinblastine, vincristine, सुना होगा, आई एम श्योर. देयर आर, so these are the first anti-cancer agents. ठीक है. So they were like it was first purified from the flower called periwinkle. जो ऐसे पर्पल व्हाइट कलर के नहीं रहते हैं, हिंदी में सदाबहार फूल कहते हैं, इंग्लिश में पेरीविंकल. You must have seen, Google करके देखना. Okay, so see the flowers from there, they actually purify, and again they are also like they bind to the microtubule proteins in order to prevent the depolymerization of the mitotic spindle and all, and hence the cells get stuck at the metaphase only. So eventually what is happening? You are preventing the cell division. Correct? So cell division prevent hoga, so obviously you can stop the cancer. And that's why it was first known as the anti-cancer agent. Anyways, now so we have got so many things which act as the anti-cancer agents, but none of it is actually helping. Okay. So tumors expressing MDR are frequently resistant to chemotherapeutic agents and thus difficult to treat. Why? As I said, जैसे bacteria के case में they were pumping out the toxin out, in the same way cancer cells were also pumping out the drugs out. Because of which the drugs which goes inside is not able to kill the cancer cell or induce apoptosis. Eventually, the cancer cell is surviving. Okay? Clear? Now, look at over here. So what is happening? The tumor survival mechanism. So these are the 6 different ways by which a tumor cell can survive. पहला क्या होगा? We can talk about each. ऐसा नहीं कि पहला ये होगा, second ये होगा. This one is responsible for your MDR. ठीक है? या ABC. Okay. So these reflex pumps, what they are doing. So if the drugs are present, these pumps will take, even if suppose by chance, if we say that मैंने एकदम lipid or having the hydrophobic drug मैंने pass किया है, because since it is going to match with the plasma membrane, it can easily diffuse the plasma membrane and goes inside of the cell. Then these cancer cells will take this drug and pump out. ये एक mechanism हो गया, ठीक है? You can do is that change the plasma membrane composition in such a way that it is going to decrease the uptake of the drug. ठीक है? ये दूसरा mechanism हो गया. Third, increase the drug metabolism. So this is what I was explaining, that you secrete enzymes which can actually detoxify the drug. Okay, so this is third mechanism. Then alter the drug targets. For example, this particular drug is there. It is targeting all the mitochondria of the cancer cells. ठीक है? अब क्या होगा? माइटोकॉन्ड्रिया नहीं होगा तो ऑब्वियसली एटीपी नहीं मिलेगा. एटीपी नहीं होगा तो सेल सर्वाइव नहीं करेगा. So if suppose a particular drug is targeting mitochondria, the cancer cells, what they can do is they can alter the path or alter the mechanism of recognizing the mitochondria. तो क्या होगा? Drug अब mitochondria को समझ ही नहीं पाएगा, so it is not going to target the mitochondria. This is fourth mechanism. Okay. Impair the apoptotic pathway. So cell does understand that कि अब मैं control में नहीं हूँ. Okay, I am not following the cell cycle ka checkpoints and I am just behaving abnormally. The cell do understand this, but to make sure that it is not proliferating continuously without any check, we have got the apoptotic proteins. Okay, cells, that is cell suicide जो हम बोलते हैं, किसको जाना चाहिए. But in the case of cancer cells, what they do is they are going to damage or decrease the expression of the apoptotic proteins. They increase the expression of the anti-apoptotic proteins. ये चीज समझ रहे हैं? So in a cell we have both apoptotic and anti-apoptotic proteins. See, apoptotic क्यों चाहिए? Because suppose cell understands कि हाँ हमारे पास अभी nutrition नहीं है, I cannot survive and maybe कुछ damage हो गया, infection हो गया. So instead of spreading to the entire tissue, the cell will understand and undergo apoptosis. But cancer cell के case में क्या होता है? They know that they are behaving abnormally, but their main motto is to survive. Their main motto is to reproduce unregulated, right? Just proliferating without any regulation. So उसके लिए what they do is they change the apoptotic pathway by increasing the expression of the anti-apoptotic proteins. For example, we'll talk about this. We'll come to that. Don't worry. Okay. And last, ये हो गया आपका fifth mechanism. Now coming to the sixth one, alter the cell cycle checkpoints. So there are actually 3 checkpoints are there. G1 में होता है, G2 में होता है, M phase में होता है. So in a cell cycle, you know that the cell cycle is divided into 2 phases, correct? Interphase and M phase, mitosis phase. Interphase में again you have got the G1, G2, S phase is there, right? Now all these checkpoints are there which ensure that the DNA is replicated or not, all the proteins are present or not. अगर DNA replication हुआ है, if there is any error, if those has been repaired or not, so all these checkpoints are there. But in the case of the cancer cell, it just overlook all these checkpoints and directly goes for the mitosis. यानी कि क्या, even if the chromosomes are not copied properly, even if there is the presence of a broken chromosomes, ठीक है, or translocated chromosomes, still it will make or it will force the cell to undergo the mitosis, and you will have the undivided, no, sorry, not equally divided cells, daughter cells will be there. So of course, since they have the cancer-based proteins more, so they are going to divide eventually by taking all the glucose from the normal cells or normal tissues. Okay, guys, following. थोड़ा जल्दी जल्दी बोल दिया, but I hope you got the point over here. So these are the different mechanisms by which a tumor cell is surviving, of which we were discussing this, the ABC transporter. Any doubts, guys? Akshaya, I know you have joined late, but I hope this is clear to you, like how, if any one tumor cell is there, what are the different ways by which it can survive?
Yes.
Yeah. Okay. Shruti, do you want to comment anything or ask anything?
No, ma'am. But there was disturbance. There was some kind of sign going on while you were talking.
I mean, so wasn't it clear? I don't know, maybe the internet, because I could see in between in my wrapper screen, it came as internet connection is unstable. So there was a time when I increased my voice. I mean,
It
what I
is still
want to say.
happening. While you're talking, it is still happening.
I guess the Wi-Fi is not stable here. So my point is, did you get the all the six mechanisms by which the tumor cell
Yes.
is able to survive? Yeah.
Yes.
And this is just the story. When we are going to talk about cancer in detail, this all we are going to do in depth. ठीक है? तो कैसे वो प्रोटीन, कैसे वो इंक्रीज कर रहा है, like how it is over expressing, how it is inhibiting, everything in detail with lot of number of proteins we are going to discuss, and of course you have to remember all of them. Okay, so now coming to the MDR mechanism, वो कैसे करेगा, ठीक है? Okay, so let us say this is your MDR. You can see that 2 ATPs are there. Okay, now if suppose it is there, passive diffusion, जैसे भी मैंने बोला, that if it is hydrophobic in nature, what will happen? It will just pass into the pass through the plasma membrane, right? Now the cell understands and it makes sure that this has been taken near to the MDR and it is pumped out. This is simple, one mechanism. Okay. Now coming to the P glycoprotein. This is P glycoprotein. Wherever if it is mentioned, then you should understand that they are talking about the bacterial MDR receptor or transporter. Okay, so PGP or P glycoprotein is present in the case of the bacteria only. Okay, this time is getting over. So now suppose over here, if suppose these P glycoprotein is there, these are your the drug or any other molecule, right? So what is happening? Can you see? ये पूरा अंदर नहीं गया, ये सिर्फ आपके आउटर में है या फिर इनर में है, फ्लिप फ्लॉप हो रहा है, करेक्ट? So what is happening? Even though it is not going inside, कैसे होगा? I'll just explain once again. So you have got this. Let us say that this is a drug. Okay. So it might go to the outer membrane, then it go to the inner membrane, and then finally going to the inside of the cell. Okay. What P glycoprotein will do? जैसे ही वो outer membrane में आ रहा है, it will take to the inner and then it will be recruited towards the P glycoprotein and so on. समझा? Usually this happens if you don't have P glycoprotein. Outer में है, inner में है, and then it comes inside. Now once you have got the P glycoprotein, outer में है, inner में है, thrown out. Yes? Clear, guys? Clear? Hello.